hydrolytic enzyme for proteolysis. So proteasome contains hydrolytic enzymes for proteolysis, and that means that the enzymes are going to use hydrolysis to break down the target protein into peptide fragments. The peptide fragments can be further broken down by the cell to individual amino acids. And this is actually the final point that I was going to make on Wednesday, talking about different points where we regulate gene expression. So regulation of gene expression goes all the way from the level of DNA structure all the way to protein degradation. All of that comes under the category of regulation of gene expression. A lot of people think in terms of gene expression as just being whether or not the DNA is going to be transcribed into messenger RNA. But that's not true. Regulation of gene expression goes all the way from opening up the DNA so it's accessible by RNA polymerase, all the way down to protein degradation. All of these encompass the concept of gene expression in a eukaryotic cell. Does anybody have a question? Okay. So what we're going to focus on today is the actual process of transcription, whereby DNA is recognized by RNA polymerase and transcribed into a messenger RNA. And what I've shown up here is the central dogma of gene expression, where DNA is transcribed into RNA. RNA is then translated into protein. And of course, we talked about the variation between prokaryotic cells and eukaryotic cells, where in a bacterial cell, transcription and translation are coupled because there's no nucleus in the bacterial cell. So as RNA is being made off of the DNA, then ribosomes bind automatically to the messenger RNA and begin translating it into a polypeptide. Translation in our cells does not happen except out in the cytoplasm. So DNA is transcribed into a pre-messenger RNA. That pre-messenger RNA is processed into a mature messenger RNA with very specific features that allow it to be transported out of the nucleus through the nuclear pore into the cytoplasm, where that RNA is going to bind to ribosomes, and then the ribosomes are going to be translating the messenger RNA into a polypeptide. So this is the overall process, or the central dogma of gene expression, right here. And as I indicated in 2 lectures before this one, for genes to be expressed, what has to happen is the DNA itself has to be open and accessible to RNA polymerase. And so as you recall, when I was talking about DNA structure, the chromosomal structure, the chromosomes are looped. The DNA of the chromosomes are looped into 300 nanometer fibers. But in these 300 nanometer fibers, you actually have loops of transcribed DNA. These loops of DNA that contain the genes that encode proteins that our cells are going to use. These loops are actually attached to the nuclear matrix. So that nuclear matrix holds up segments of the DNA so that it can be accessed by RNA polymerase. And a nice little diagram is shown here. It doesn't show RNA polymerase coming in, but actually these loops of DNA are actually held up by proteins in the nuclear matrix to allow genes on these looped out segments to be accessed by RNA polymerase. And this is an electron micrograph showing, well, it doesn't show it very well, but nonetheless, this is the scaffold protein they were referring to, and the loops of DNA accessible to RNA polymerase. And so if we look at the nucleosome, if you recall, the nucleosome is an octet of histones, 2 of histone H2A, 2 copies of histone H2B, 2 copies of histone 3, 2 copies of histone 4. These are 2 copies of each of these histone proteins form an octet, around which 140 base pairs of DNA wrap. And there are two ways that the chromosomal structure can allow for RNA polymerase to come in. First, the DNA cannot have any 5-methylcytosines in the promoter or enhancer regions. Remember, 5-methylcytosine is the only modification known in mammalian DNA. And what 5-methylcytosine does is it extends down into the major group of the DNA. It prevents transcription factors from binding. But what it also does is it encourages the binding by what we call DNA deacetylases or histone deacetylases. And if you recall again, the histones H3 and H4 have acetyl groups attached to their tails. And by binding to acetyl groups on the tails of histone H3 and H4, the positive charge that allows for tight histone binding to negatively charged DNA, those positive charges are partially blocked, and therefore the nucleosome is a little bit more loose. And this allows for an RNA polymerase to come marching through. And so by virtue of acetylation of histone H3 and H4 tails, as well as methylation of histone 3, lysine 4, but also there's no 5-methylcytosine on those promoter or enhancer regions, and that's what allows for DNA to be open and available to RNA polymerase. When DNA is not open and available to RNA polymerase, there's hypoacetylation of histones H3 and H4. Hypoacetylation removes these acetyl groups. Therefore, the positive charges on the histone tails are now available, and they bind very tightly to the negatively charged DNA. And that actually binds up the DNA and the histone proteins very tightly. And it begins the process of formation of what we call heterochromatin. Heterochromatin is another way of saying inactive chromatin. Heterochromatin is inactive chromatin. In other words, it's not accessible to RNA polymerase. Another modification that distinguishes heterochromatin is methylation of histone H3, lysine 9, as opposed to lysine 4. This is actually called the histone code. And the histone code and its role in whether or not DNA was accessible to RNA polymerase, this has only been worked out in the last 25 years or so. It's called the histone code. Prior to that, the only thing that was known was that it seemed like cytosine methylation had a strong effect on gene expression. But it was not, and it was known that that wasn't the only thing. So this is the histone code, and that also, of course, determines whether or not genes are going to be expressed. Can I repeat anything? Is this understandable, or at least somewhat clear to you? I think I've probably described the process how many times now? 5? So you should be okay. If you're not, please let me know. I'm happy to answer any questions. All right. So now we're going to talk about the process of transcription proper. So, of course, our DNA that's bound up in our chromosomes, along the stretches of our DNA, we have genes that encode proteins or ribosomal RNA or transfer RNA or microRNA. And what happens is during transcription, the RNA polymerase will bind to a strand of the DNA that has So what happens is DNA polymerase binds to the coding strand 3', and then in an anti-parallel fashion generates messenger RNA 5', 3'. So the template strand is in a 3' to 5' orientation. The newly synthesized messenger RNA is in a 5' to 3'. So DNA to RNA, these are still in anti-parallel orientation, okay? Very much like the two strands of DNA that wrap around each other, that how they're in anti-parallel orientation. Well, DNA as a template and messenger RNA, they're also oriented in an anti-parallel fashion. All right? Okay. So what happens when the DNA is transcribed by RNA polymerase is you get a messenger RNA. When it comes time to translate that messenger RNA, the messenger RNA is read by the ribosome in what are called triplet codons. And each one of these triplet codons is going to be translated into an amino acid. And we're going to spend quite a bit of time, we're going to spend a whole lecture on the process of translation. What we're going to do today is focus on transcription. But this gives you an overall picture of how the process happens. Okay, so there are 4 known gene categories. Messenger RNA or mRNA are RNAs that encode proteins, and they're transcribed by RNA polymerase II. So messenger RNAs are transcribed by RNA polymerase II. The microRNAs that I talked about in our last lecture, those are also transcribed by RNA polymerase II. Ribosomal RNA. Ribosomal RNA, as you recall, is actually generated in a segment of the nucleus called the nucleolus. Remember that? The nucleolus is where we have RNA polymerase actually bound to the ribosomal RNA genes and generating lots of ribosomal RNA. It forms a little organelle called the nucleolus. That's where you find RNA polymerase I. And then, most of our genes, interestingly enough, most of our genes are scattered all throughout our genome. But it's interesting how ribosomal RNA genes, as well as tRNA genes, oftentimes come in clusters. And the RNA polymerase that transfer RNA genes is RNA polymerase III. Okay. Any questions? Are we okay? Your book really doesn't go into this. So this is important for you to know to be able to distinguish what genes are transcribed by what. All right, what I'm showing here is the known genetic code. And so for these triplet codons that are present in our messenger RNAs, as I indicated, each one of these triplet codons is going to be translated into a separate amino acid. What's interesting when we look at the genetic code is there's quite a bit of redundancy. There's a lot of redundancy. For example, there are multiple. I can see that there are six different types of codons that can encode for the amino acid serine. There are, let's see, when it comes to valine, we are housekeeping genes, or genes that simply encode proteins critical for the life of all cells, that those genes actually are driven simply from a promoter. But we also know, and I've described this to you in previous lectures, that genes that are what we call tissue specific, that give specific cells in our tissues a distinct identity. Tissue specific genes are driven by cancers that can bind to tissue specific transcription factors called activators. And I also described how if you have a tissue-specific gene that's being transcribed, this enhancer is going to bend to encounter the promoter region of the gene, and it does that through DNA bending protein. And this allows for general transcription factors that bind to the promoter to come in contact with tissue-specific transcription factors that are bound to the enhancer. These factors then establish what is called a transcription initiation complex that allows for RNA polymerase, whether it's RNA polymerase 2, polymerase 1, or polymerase 3. These factors will allow for RNA polymerase to bind. And if it's RNA, it doesn't matter what kind of RNA polymerase, you have that transcription initiation complex. RNA polymerase binds at the promoter region of the gene. It then starts to march along the transcription unit, generating messenger RNA. And again, what will happen is the polymerase will actually bind, and it will actually start to read. It will read the gene 3' to 5', and generate from it a 5' to 3' messenger RNA. And the process where the RNA polymerase marches along the length of the gene is called elongation. And then, when the RNA polymerase hits the end of the gene, the RNA polymerase will actually fall off, and that's called termination. Then you have a completed RNA transcript generated, like I said, 5' to 3'. This is shown in a little bit more detail right here. So in the process of elongation, what is happening is the RNA polymerase is actually reading the template strand 3' to 5' to generate a messenger RNA in a 5' to 3' direction. So anti-parallel to the template strand. And the RNA polymerase, what does it do? It actually inserts a base across from the template DNA. So when RNA polymerase encounters a thymine, it will insert adenine in the messenger RNA. When the RNA polymerase encounters an adenine, it will insert a uracil across from the adenine. And that's because RNA, remember, doesn't contain thymine. It doesn't contain thymine, it contains uracil. When RNA polymerase encounters a guanine, it will base pair the cytosine across from it. And that's how the polymerase reads the template DNA. Does anybody have a question? What I want to stress also is that whether it's a housekeeping gene that simply is driven by a promoter, or a tissue-specific gene that's driven by an enhancer and a promoter, what's interesting about what we call eukaryotic promoters, or the promoters of the genes in our own cells, they have a sequence, TA, TAAAA, it's called the Tata box, invariably in regions. What's interesting about the Tata box is that it's a series of AT base pairs. What do we know about AT base pairs? That segment of the DNA is not held as tightly together as you would see with CG base pairs. Remember, CG base pairs have 3 hydrogen bonds helping to hold the helix together. With AT base, a stretch of ATs, the DNA is not held as strongly together, and it helps to make these Tata boxes a little more easy to open by the RNA polymerase and the transcription initiation complex. Okay? So what happens is the transcription factors will bind over the Tata box, whether they're tissue-specific or general transcription factors. Once these factors have bound over the promoter, they recognize the Tata box. Other transcription factors can then be involved. And finally, RNA polymerase 2 will bind start to elongate the messenger RNA, like I said, in a 5' to 3' direction, until it hits what is called a termination signal. At the end of the gene, there will be a termination signal, and that's when the transcription complex just simply falls off of the DNA. Okay. Does anybody have a question? All right, so what I want to point out now is what do these transcription factors look like? Well, we know that transcription factors, most of them have the ability to bind to DNA, otherwise. And so, consequently, most transcription factors have what is called a DNA binding domain. Transcription factors also have what is called an activation domain. And this is a domain or a segment of the transcription factor that allows it to be activated. Do you remember when I was talking to you about cell signaling pathways and how in cell signaling pathways there is a kinase that will go from the cytoplasm into the nucleus to activate transcription factors to make them bind to the DNA and start reading the genes? Well, that would be the activation domain, the segment of the transcription factor that becomes phosphorylated. And then another thing that transcription factors will frequently have is a protein-protein interaction domain. Because as you can see in the diagram here, transcription factors aren't acting on their own. They interact with other transcription factors. They interact with other transcription factors to form an initiation complex. And so transcription factors have a DNA binding domain. They have an activation domain where they can be phosphorylated and activated from a kinase that migrates from the cytoplasm into the nucleus. And then they have what is called a protein-protein interaction domain. And domains in proteins are just a segment of the protein that carries out a certain type of activity. Am I clear so far? You okay? All right. And I've given you this before, but it must be really important for me to give it to you again. And that is for a messenger RNA to be stable and be exported from the nucleus out into the cytoplasm so it can be translated. That messenger RNA has to have a 5' 7-methylguanine cap. It has to have a poly-A tail. And another thing that messenger RNA is going to have is a 3' untranslated region. So the messenger RNA that's generated from the DNA template is shown here. What's interesting is the messenger RNA will have a start codon that encodes for methionine. It'll have a stop codon. So the start codon is where the ribosome will actually bind to the messenger RNA and begin translating. The stop codon is where the ribosome is actually going to come to a halt and it's going to fall off of the messenger RNA. And then what you'll have is a polyadenylation signal. And in eukaryotic cells, the polyadenylation signal that is built into all of our RNAs is AAU, AAA. And this is the signal that is recognized and bound by poly-A polymerase, which generates this poly-A tail. Any other questions? Are we okay? All right. What's also important, and your book doesn't go into this, but I can tell you for sure, this 3' untranslated region actually is recognized and bound by a series of RNA binding proteins. So there are RNA binding proteins that bind to this 3' untranslated region. They're also very important in keeping the RNA from being degraded. So all of these features, the 5' cap, the 3' untranslated region with with RNA binding proteins as well as the poly A tail. All of these are critical for maintaining the messenger RNA so it exits the nucleus, go out into the cytoplasm, and be read by the ribosomes to generate a polypeptide. Okay? So I'm going to spend a few minutes on the concept of RNA splicing. If you recall in our last lecture, I talked about how it was initially thought that our genome would have over 100,000 genes, but everybody was so disappointed that there's only 25,000 genes in our genome. That's not a whole lot, and there's not but 2% difference in our genome compared to a chimpanzee genome. So, oh my goodness, what is hidden in our genome that allows us to be us and distinguish us is that we have different types of what we call tissue-specific transcription factors that bind to enhancers. We also have what are called tissue-specific splicing factors that are involved in determining which of these introns is actually spliced out of the pre-messenger RNAs. So there are 2 points where tissue-specific factors are regulating gene expression. First, when tissue-specific transcription factors bind to the enhancers, and then when tissue-specific splicing factors help to determine which introns are actually going to be spliced out of the messenger RNA. And in the process of splicing, what happens is we have what is called a spliceosome. It's a complex of small nuclear ribonucleoproteins, small nuclear ribonucleoproteins, and that actually is small nuclear RNAs as well as spliceosome proteins. And what I want to point out is it's the RNA in the spliceosome, the RNA in the spliceosome actually forms base pairs. It forms base pairs with the messenger RNA at the intron sequence. So the spliceosome itself involves a small nuclear ribonucleoprotein complex. That's what this is. Small nuclear ribonucleoprotein complex that uses the RNA, or small nuclear RNA, to form base pairs with the messenger RNA at the 5' part of the intron and also at the 3' portion of the intron. And that's what you're looking at here. That's what you're looking at here. This is actually a simplified diagram of what's called a spliceosome, which is a series of small nuclear ribonucleoprotein complexes that bind to the intron sequence of a messenger RNA. This is then called the spliceosome. And what I want to stress is where the introns are distinguished by the spliceosome is at the GT and AG sequence. So always in eukaryotic messenger RNAs, what distinguishes an intron is a GT sequence at the 5' portion of the intron and an AG sequence at the 3' portion of the intron. And the GT and AG are actually recognized by the small nuclear RNAs in the small nuclear RNA ribonucleoprotein complex. And they're part of the proteins that are involved here are actually ribonucleases that will cleave the phosphodiester backbone of the RNA. The exon will be spliced out, and then the 2 ends of the exons are pulled together and sealed. And again, the ribonuclease that cuts the RNA and the enzyme that reestablishes a covalent bond in the phosphodiester Phosphodiester linkage is also a protein that's a part of the spliceosome. And then you get what is called the mature messenger RNA. Can I repeat anything or explain anything again? Are we okay? Question?
So the GT.
Alternative splicing to actually generate all kinds of what we call messenger RNA isoforms. So a single gene with many, many different introns and exons can be alternatively spliced to give what we would call protein isoforms. And some of these protein isoforms look very different, and they all come from the same gene. And that's how complex organisms such as ourselves can live along on what seems like a pretty paltry 25,000 genes. It's through alternative RNA splicing that our genome is maximized. And an example is the alpha-tropomyosin messenger RNA. It's an alpha-tropomyosin, so it's a form of a motor protein, and that's you find in muscle, and also other types of cells. So, for example, the splicing pattern that we find in striated muscle is shown here. The splicing pattern from the alpha-tropomyosin messenger RNA that we find from another type of striated muscle is shown here. But you can get a completely different splicing pattern in liver cells, in non-muscle fibroblasts, so these would be the cells underneath your skin, as well as in your brain, your neurons in your brain, for example. And so that's how you get different types of proteins from the same gene, is by alternative splicing. And so these tissue-specific splicing factors that I just talked about, the tissue-specific splicing factors that determine which intron and which exon are going to be retained or spliced out, those tissue-specific splicing factors are also part of the spliceosome. They would be proteins that are actually involved in this small nuclear ribonucleoprotein complex. These proteins are involve general splicing proteins, as well as tissue-specific splicing factors that would give rise to the different splicing that you see from the same pre-messenger RNA. Okay? So do we understand that concept? Your book doesn't talk about it too much.